The overall goal of this atomic force microscopy experiment is to resolve interactions of DNA repair proteins with damaged sites in DNA, and to distinguish between different DNA interactions by various protein complexes.
This method can help answer key questions in the DNA repair field, such as how a particular DNA repair system achieves the recognition of its target lesions in the DNA. The main advantage of this technique is that we can directly see and analyze the different types of complexes that are involved in the DNA lesion recognition process. Demonstrating the procedure will be Nicolas Wirth, who used this approach during his bachelor research.
To begin, prepare a suitable DNA substrate by generating a single-stranded DNA gap in a plasmid. Refill the gap with modified single-stranded DNA oligonucleotides. And finally, linearize the plasmid DNA using appropriate restriction enzymes.
For these experiments, the double-stranded DNA substrates are about 1,000 base pairs long and contain a DNA lesion, as well as an unpaired region called a DNA bubble for protein loading onto the DNA.
Next, prepare a protein DNA sample for AFM analysis by first pipetting 10x reaction buffer and water into a microreaction tube. Then, add the DNA and proteins under investigation. For example here, 100 nanomolar of the DNA sample and 1 micromolar of each of the eukaryotic nucleotide excision repair proteins XPD and P44. Then, incubate the reaction for 30 minutes at 37 degrees Celsius in a heat block.